to my cutest little champions. I welcome you all to study sexual reproduction in flowering plants with me, my dear children, in one go. And I welcome you all to Masterstroke series, which is exclusively made for KCET aspirants. But let me tell you, someone else is also going to get the benefit of it, and that is PU2 board students. Because as of now, we are covering the PU2 chapters in one go. So if you are thinking that I do not have much time for my boards left and I want to have a quick revision video, this is perfect for you. Just go through it. And obviously, I have included just the most important points over here, so I don't want you to have a pen and paper while you are going through this particular session. I just want you to have your NCERT so that you can see that whatever ma'am is teaching is coming from here. You get it? Fine. And now one more very important thing. That is, if you are ready to study this chapter with me, then comment down that we are ready to study. Well, that's not a very big punchline, but yeah. So, jokes apart, let's get into the chapter. Now, the first part of the chapter, in that it is said about the male reproductive system. And what do you call this male reproductive system as? You call it androecium, my dear children. Now, what does androecium contain? Androecium contain a long filament, and on the top of the filament you have a structure like this, which is known as anther. Together, what do you call it? You call it stamen. But ma'am, you told that it is known as androecium. So bacha, it's the other name of androecium, that is stamen. Now, the anther is something which is like the hero or heroine of the story. So over here we have to talk a lot about anther. So let's see the structure of anther. I say anther is bilobed, as in it has two lobes. One lobe is this, another lobe is this. Now, each lobe that you see over here is divided into two theca. So over here, if you see, they are divided into two theca. This is also divided into two theca. So I'll call it dithecus. Now there's a longitudinal groove that runs along with it, dividing them into what, my dear children? Two theca. Okay, now the anther is four-sided. It is four-sided. 1, 2, 3, four-sided is there. Yeah, and now comes the most important thing up over here. The anther, each theca of the anther, it contains microsporangia. So basically, there is one microsporangium here, 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 here. And what are microsporangium? They are going to be the factories of pollen grains. So they are going to go act like pollen sacs. This is where the pollen grains are going to be made. They have not been made yet, but they will be made. And how does the structure look? So basically, if I say this is one theca, this is another, this is another, this is another. So each of them, I will say it has microsporangium. Now, microsporangium has its own structure. Let's see. So the innermost thing which is there over here, I will call this as sporogenous tissue, which is there. Now this is found in the center, later forms microspore. So this sporogenous tissue which is there, this is a tissue. It is made up of so many cells, and these cells are the ones which would be dividing and giving rise to pollen grains later on, which is why I call the whole structure of microsporangium as what? Pollen sacs, my dear children. Okay, now going ahead, there is someone to provide nourishment to the sporogenous tissue, and that is what, my dear children? The tapetum. So the layer outside the sporogenous tissue, it will be known as what? Tapetum. And what does tapetum do? It nourishes the developing pollen grains. There are certain things that you should know about tapetum. That is, it has very dense cytoplasm because obviously it has to provide nourishment, and often it is binucleate. That means in one cell you will be finding two nucleus. Why it is happening? Probably because usually when the nuclear division happens, after which the cell also divides. But probably here, after the nuclear division has happened, the cell might not have divided, which is why both the nucleus are there in one particular cell. So sporogenous tissue, outside it you have tapetum, which provides nourishment. Okay, now outside the tapetum you have three layers. Outside the tapetum you have three layers. What are those? Epidermis, endothecium, and the middle layer. Okay, epidermis, endothecium, and the middle layer. So three outer layers which are there, my dear children. What is their use? So tapetum provides nourishment. But what is the other uses of all three layers? My dear children, they provide protection and helps in anther dehiscence. So the pollen grains are being made up over here. How do they release? Basically, for that the anther has to devise. They have to break open and release all the pollen grains. So for dehiscence of anther, what do we have? We have all these three layers, and also for providing protection as well. So I hope this is completed. Now this is a very important structure that you have to remember. So epidermis, inside that you have endothecium, and then you have middle layer, and then you have tapetum. Inside the tapetum you have the sporogenous tissue. The sporogenous tissue, my dear children, I told you, each of them has an ability to give rise to microspores. to give rise to microspores. So what do I call these cells as? I would be calling this cells as MMC, microspore mother cell. So because each of them has an ability to divide, I will be calling it microspore mother cell. Okay, going ahead, talking about the formation of pollen grains. How are it formed? So I told you in the sporogenous tissue, all the cells can divide, right? So I'll take one cell. This is a microspore mother cell, okay, which has diploid number of chromosomes. Obviously, diploid number of DNA, okay? The amount of DNA is diploid. Now, as in the normal cell. Now, it will be undergoing some process which is known as meiosis, and meiosis happens in two phases. Meiosis 1 is there, well, the cell will divide, but it will have the number of chromosomes. So, it has half the number of chromosome up over here in meiosis 1. Again, what will happen? There will be meiosis 2. In meiosis 2, again it will be dividing, but there won't be any halving. Whatever thing is there, it will be passed down over here. So meiosis happens like this, where one cell will be dividing into 4 separate cells, my dear children. So this was diploid. The microspore mother cell was diploid. Over here, you got haploid cells, and then later they divided. Again, you have 4 haploid what? Microspores. You have 4 haploid microspores. 4 of them initially they are together, later, okay, later because of dehydration and lot of things, they will be separated, and when they are separated, you call each of them as pollen grains. What do you call them? Pollen grains. So sporogenous cells basically each can form microspore tetrad. Now microspore tetrad as in this thing 1, 2, 3, 4 tetrad. Now microsporogenesis, meiosis in pollen mother cell happens, or basically you can say microspore mother cell. Either way, it's the same thing. Now produces microspores. Microspore tetrad is initially grouped in 4, but later they separate. Maturation, anther dehydrates, microspore deassociates and develop into pollen grains. Then the pollen release is there. Thousands of pollen grains are being released when the anther it dehisces. Now we are going to talk about the structure of the pollen grains. Why have we released the pollen grains? Because my dear children, pollen grains will be having the male gametes in them. Pollen grain is not the male gamete. Okay, pollen grains are the one that produces male gametes. So the one that produces male gamete, what can I call it? I can call it male gametophyte. I will be calling it male gametophyte because it is going to form the pollen grains, the gametes. Now over here talking about the male gametophyte, you see the structure. So I will tell you the structure. So over here you can imagine there is a discontinuous layer, that is outer layer. You call this as exine, and then there is a continuous layer which you call as intine, my dear children. And why is this exine incontinuous? Because here you see pores. What do you call this pores as? Germ pores. Now this is intine, and then there is your pollen grain with its own plasma membrane. With its own plasma membrane. Now I'll tell you one thing. The pollen grain, majority of it is basically a cell which is known as vegetative cell. What is it known as? Vegetative cell. Now the vegetative cell, no, inside the cytoplasm of it, it harbors a small cell which is known as generative cell. This is what? Generative cell, my dear children. This generative cell later will be undergoing division to give rise to 2 gametes. It gives rise to what? 2 male gametes. So each pollen grains, my dear children, has a capacity to produce 2 male gametes from the generative cell. What does the vegetative cell do? It provides nourishment to the generative cell over here. Okay. Now, why do you have germ pore? Basically, the pollen grain, when it lands on the stigma, okay, of the female plant, okay, what will happen? This pollen grain has to form a pollen tube, and this process is known as germination, pollen germination. So for pollen tube formation, obviously you need a space, and for that you have germ pores, okay, and also for a little bit of nourishment and stuff like that. Now the exine which is there, it is made up of a very tough substance which is very, very important. Its name is sporopollenin. Its name is sporopollenin, which will be able to undergo, which will be able to survive any type of situation. Whether it's alkaline, acidic, it's too hot, too cold, whatever is there, it will be able to survive. So let's go through all the points that I have told you. Then there are certain extra points that I will be telling you. Now, male gametophyte, that is pollen grains, represent the male reproductive units. I told you In male gametophyte, it's the pollen grains. Now, observation. You'll be seeing variety in size, shape and color design under the microscope. Size is very much important. It is 25 to 50 micrometers in diameter. What is it? 25 to 50 micrometers in diameter. Talking about the wall layers, there is exine, my dear children, made up of sporopollen, which is a very tough substance, highly resistant material. Please remember, very important question. Now it has germ pores also, which is why it is not continuous. Now talking about in time, the inner layer, it is made up of cellulose and pectin. What is it? Cellulose and pectin. Talking about cytoplasm, it is enclosed by the plasma membrane. As I told you, in the cell you have vegetative cell that is larger, stores food and has an irregular nucleus. Please remember the structure of the nucleus. What is it? Irregular nucleus. Talking about generative cell, which is there, it is small, it is spindle shaped and floats in the vegetative cell. Here the shape of the nucleus is also kind of spindle shape. Cool. Now talking about the pollen shedding, I told you that the generative cell can divide into two male gametes. So usually, okay, only in 40% plants, that is angiosperms, flowering plants, it is divided like that. For example, vegetative cell will be there obviously, and then the generative cell divides into two. So how many cells do you have in total? 1, 2, 3. So I'll call that this particular stage is 3-celled stage. So 40% of the plant shed at 3-celled stage, that is there will be a generative cell that will be divided into two male gametes. But 60 more of it, okay, more of it, 60% of the plants, my dear children, are just going to divide, are just going to basically shed in, okay, shed in 2-celled state. That is, there will be vegetative, there will be generative. The generative would not be dividing until and unless it goes and germinates. So until and unless it will be coming and landing on the stigma, it won't be dividing. So this is more common. That is why 60% of the plants show that. Cool? Works. So all about pollen grains is also done. Talking about pollen as an allergen and also a performance booster. So there is nothing to explain over here. You have to understand this. Pollen allergies, obviously it's there. It causes asthma, bronchitis, respiratory disorders in some people. If we talk about allergies, you know Parthenium. Parthenium is carrot grass and it was an invasive weed causing severe pollen allergies in India. Now nutrient rich, the pollen grains, obviously as you know that generative cell has to be dividing into male gametes. So vegetative cell provides a lot of nourishment. So obviously there are high amount of nutrients in it. So pollen grains are highly nutritious and can be used as food supplements also. There are pollen products. You get tablets and syrups as well, and it is very popular in Western countries. Now another very important point is that it is going to act like performance booster. It is claimed to enhance the stamina in athletes and racehorses. Cool? Now, there is something that is known as pollen viability. This pollen should be having the ability to go and germinate. So how long do you think the pollen grain can be viable? Let's see that. So pollen must land on the stigma before losing its viability for fertilization. Viability varies depending on the temperature and the humidity. There are short-lived pollen. You have to remember the names. It is rice, wheat. That will lose its viability within just 30 minutes. Rice and wheat, the staple food that we eat, my dear children, it loses its viability within just 30 minutes. What about long-lived pollens? There are 3 families that are given: Rosaceae, Leguminosaceae and Solanaceae. It is viable for months. So this is contrasting. Here it is just for 30 minutes they are viable. Here they are viable for months. Okay. Now you have pollen storage. So it can be stored in liquid nitrogen minus 190, minus is there. So minus 196 degrees Celsius. That is the temperature where it is stored. Okay, why in this temperature? Because in this temperature there is no freezing happening. Okay, there is no ice crystal formation. Not, I won't say freezing happening. Basically there is no ice crystal formation happening. In lower temperature obviously crystal formation happens, but it is going beyond it. Okay, so we are keeping in minus 196 degrees Celsius in liquid nitrogen, my dear children. And then this pollen grains basically can be used for crop breeding programs and seed banks and things like that. And with this, we are going to complete the male reproductive system. Getting ahead with the female reproductive system. The female reproductive system, what it is known as? It is known as, in simple language, pistil, or you call this as carpel. What do you call it? Pistil, or you call it as carpel. Now pistil, there are types. There are monocarpellary, that means there is only single pistil. Okay, multicarpellary, that means there will be more than one pistil. Now there is condition, syncarpus and apocarpus. Syncarpus, as you can see over here. Okay, here the carpels, all of them, they are together. Okay, so you call this condition as syncarpus. And what is an example for this? Example is papaver. Okay, papaver is an example of a syncarpous plant. Please remember it. Below this picture diagram, there is in the description box it is written. Okay, now apocarpus, as in the pistils are free. As you can see over here, this is 1, this is 2, this is 3. So where do you see this? You see this in Michelia. Again, given in the diagrams, overview. Okay, you have to go through it. Now, if you talk about the pistil structure, general structure, my dear children, there will be stigma. Okay, there will be stigma, and where will I make it? I can make it here. So there is a landing surface that is known as stigma, and then there is a style, and then there is what? An ovary. Inside the ovary you have an ovule, and then inside the ovary you also have placenta. What do you call? Placenta, which will be providing nourishment to this particular ovule. Why is it providing nourishment to the ovule? You'll get to know that. So stigma, style and ovary is there. Now a basal swollen part contains ovarian cavity. So ovarian cavity which is there, it is known as locule, my dear children. Placenta located inside the ovarian cavity. So as you can see, this is the ovarian cavity. Inside it you have the placenta. Now ovules which are there over here, my dear children, they are known as megasporangia. Okay, they are known as megasporangia. They arise from placenta inside the ovary. So in the case of males, inside the anther you had microsporangia. Here the ovule is going to act like megasporangia. Now there will be single ovule as in wheat, paddy or mango. Okay, or there can be multiple ovules as well as in papaya, watermelon and orchids. Cool. Please remember the examples: papaya, watermelon and orchid. PW. Oh, okay. So there is no reference to any coaching center, but still. So wheat, paddy and mango, and then papaya, watermelon and that is multiple ovules are there. Here only single ovule will be there. Going ahead, if you talk about the structure of megasporangium, what do you mean by that? You are basically talking about the structure of ovules, my dear children. So what do you have over here? So I will make the structure of the ovule and it will be more easy for you to understand then. So listen to me carefully. So suppose this is the ovary that I am having. Okay, over here I have the placenta. Now over here I have the ovule. Okay, now inside the ovule, outside the ovules, my dear children, it is covered by two integuments. One is the inner integument and then there is an outer integument like this. And then there is a structure which will be providing nourishment, and this is known as funicle. This is what? Your placenta. The point where it attaches, it is known as hilum. Okay, this is outer integument. This is inner integument. And what? This area that you see, this is micropylar region. Wherever there is an opening, just understand it is a micropylar region, and the opposite end of it, my dear children, the opposite end of the opening will be always chalazal end, chalazal region. Now inside this you will be having so many cells, and this becomes a part of something that is known as nucellus. Nucellus. So just like how in the anther you had the microsporangium, in the microsporangium you had sporogenous tissue, similarly nucellus is over here, but here not all the cells are going to give rise to what? The male gametophyte. There it was producing male gametophyte. Here I am talking about female gametophyte. Not all of them have an ability to produce female gametophyte. Only one cell will be there, my dear children. Only one cell which I will be calling as, again, MMC, but this time the M stands for megaspore mother cell. So megaspore mother cell, which is 2N, my dear children, diploid. Please remember the ploidy. It will divide. Meiosis 1 is happening. So there are two cells, both of them will be haploid. Again it divides, two cells, both of them, all of them haploid. In the case of them, all of them were microspores, but over here they are megaspores for sure. But three of them are going to degenerate. Only one cell will be left behind, and this is going to form the female gametophyte. This is going to form the female gametophyte, which is known as the embryo sac. Now if you're ready, I'm going to teach you something else as well over here itself. So Some cells of the nucleus will be still persisting over there, and inside it, what do you have? You have the embryo sac being formed right now. And this embryo sac, obviously it will have a nucleus. This nucleus is going to undergo division, that is nuclear division. First nuclear division happens, there are two nucleus which are being formed. Another nuclear division happens, from here you get 2, and from here also you get 2. Okay, so I am removing the older one. Again, oh my god, okay. One more nuclear division is going to happen. This will divide into 2. This will also divide into 2. This will also divide into 2. This will also divide into 2. 1, 2, 3, 4, 5, 6, 7, 8 nucleus is being formed. How many nuclear division is happening? First, 2 and 3. So 3 nuclear division happens in the embryo sac, my dear children. And basically, initially they are not covered by the cell wall and things like that. So they are just nucleus which is dividing. There is no cell wall formation. Slowly there will be cell wall formation, and I'll tell you what is going to form. So it is a female gametophyte. It has to form the female gamete. So the first thing that it forms is the female gamete right over here. That is the egg. Okay, now surrounding the egg you have 2 cells which are known as synergids, and they have some apparatus like filiform apparatus. They have some apparatus, that is filiform apparatus. Why is it? Because the pollen tube, when it lands and come over here, okay, you need to direct the pollen tube that, hey, you have to meet the female gamete. So for that you have the filiform apparatus of synergids which will be helping it guide and getting infused with the female gamete, that is the egg. Okay, so that is the egg apparatus. All 3 cells together, you call this as the egg apparatus. Now, opposite side of it, what is happening? There are 3 other cells. Okay, and you call them antipodal cells. You call them antipodal cells. Now, how many nucleus are still being remaining? 2 more, 6 and 6. 8 nucleus was being formed, right? So 2 nucleus, my dear children, they just stay over here. They do not have any cell wall formation, things like that. Basically, both of them are in a zone, something like this. They are in a very big cell which is known as central cell. You will call this a central cell which has 2 polar nuclei. Always remember, in the micropolar region only the egg will be there. Okay, cool. So this is all about the female thingy, but let's read it for making you understand. Now over here, this is what an inverted ovule. Okay, so usually we found that the placenta was over here. Okay, which is why the funicle and everything towards down, but probably here the placenta is up, which is why the funicle is here, and the micropolar and the funicle region is here. So always understand, if micropolar is here, if the opening is here, chalazal will be still here. Don't go find funicle and write chalazal end over there. Please remember that thing. Cool. So funicle is there, that is a stalk-like attachment attaching ovule to the placenta. Hilum is a junction between the funicle and the ovule body. Integuments 1 or 2 protective layers, leaving a small opening that is known as micropyle. Micropyle is a small opening at the tip of the pollen entry for the pollen entry. Chaliza is the opposite end of the micropyle, and nucellus is what? Master cells with reserved food, just like sporogenous tissue. Going ahead, megasporogenesis, which we just studied. So formation of megaspores happens from MMC, that is megaspore mother cell, okay, found in the micropylar region of the nucellus. Micropylar region of the nucellus, there it happens. Now it undergoes meiosis, producing 4 megaspores, but 3 of them will degenerate, only 1 will be there, and hence you call this as monosporic development, because only a single functional megaspore is there that forms the embryo sac, that is the female gametophyte in the females. Cool. Going ahead, the female gametophyte, as I said, functional megaspore, nucleus divides, then you have 2 nucleate stage, again it divides 4, and then again it divides 8. So how many nucleus you have? Total 8. Now no immediate cell wall formation happens, so it's a free nuclear division. After 8 nucleate stage, cell worms form, organizing the embryo sac. So distribution, let's see, the egg apparatus is there, where 1 egg cell will be there and 2 synergids are there. 1 egg cell and 2 synergids will be there with their filiform apparatus. Okay, now there will be a central cell, contains 2 polar nuclei that you can see over here. Then there will be antipodal cell at the chalazal end. Okay, if you see at the end, how many cells you have? 1, 2, 3, 4, 5, 6, and this whole thing, that is 7 cell. But how many nucleus you have? 1, 2, 3, 4, 5, 6, 7, 8 nucleus you have. So you call this condition as 8 nucleate but 7 celled condition. This is the condition of the female gametophyte. Works, and with this we complete the female part as well. Now we are going ahead with something that is known as pollination. Obviously the pollen grains that we have made has to come and fuse with the the female gamete, and for that you need pollination. What do you mean by pollination? The transfer of pollen grains from the anther to the stigma. Don't go and write the fusion of male and female gametes. It is known as fertilization. Pollination means just you are transferring the pollen grains from the anther to the stigma. That's all. Now you have different types of pollination. One is autogamy. What do you mean by autogamy? Pollen from the anther to the stigma, the same flower. Suppose this is a flower, that is what we are having. Here you have the female part and the male part. So from the male part it comes and lands to the female part. My dear children, you call this as autogamy. But what do you exclusively need over here? Synchrony in pollen release and stigma receptivity. The pollen release should be synchronized with the stigma being receptive. Suppose the pollen is being released but the stigma is not receptive. Do you think things can work? No. So autogamy is basically self-pollination. Now under autogamy in your book there is an example given of oxalis, viola and commelina plant. So in this plant, my dear children, you get two type of flowers. One is chasmogamous flowers and the other one is clistogamous flowers. Clisto ones are near to the down area and chasmogamous are upwards. Okay, so what is chasmogamous? Chasmogamous are the open flowers. So it's just open and it's there. So obviously from other pollen grains also pollen can come, and from other plants also it can come and land. So it will be showing self as well as cross pollination. It will be showing self as well as cross pollination. So if self is happening, then basically it's the same genetic material. It's the same parent producing the male gamete and the female gamete. So genetic material is not being esteemed very much. So there is no genetic variation being seen. But if it is a cross pollination, obviously you can see lots of genetic variation. So chasmogamous flowers show self as well as cross pollination. Talking about clistogamous flowers which are there, they are closed, my dear children. So inside this, suppose this is a closed one. Here you have the female reproductive system and the male. Obviously the male cannot go anywhere, it will be releasing the pollens over here itself. So this will be obviously always autogamy. There is no other option that occurs over here. So they will be showing only autogamy. But what is the good thing about it? The advantage is that the seed production is guaranteed. Every time there will be short seed set. Okay, a short seed set that is there. Okay. Now, you do not need pollinators and everything. Now, disadvantage is that there is no genetic diversity because it's the same plant that basically you are using. Okay. Now, this is about autogamy. You have two more type of pollination. One is geitonogamy and the other one is xenogamy. What do you mean by geitonogamy? That means, for example, over here, this is kind of self-pollination only, where you have the female and the male gametes in one flower, and in the other flower, you again have it. Okay, so from here the pollen grains are going to land on the other flower but on the same plant. And suppose that is not the case. Here there is male and here there is female. From here, the plants, okay, from here things are coming over here. This is also the same plant itself. This is known as geitonogamy. Stigma of another flower on the same plant. Another flower but on the same plant. This is known as geitonogamy. So functionally it is, it's not self-pollination. Functionally it is cross-pollination, but genetically if you see it is autogamy. Okay, functionally it is cross-pollination because two different flowers are being used, but genetically if you see it's autogamy itself. The same genetic material will be crossed. Xenogamy is when you have one plant over here, another plant over there, my dear children. From here to here things are going between two different plants, obviously two different genetic material. So this is known as xenogamy, and this is true cross-pollination and provides more genetic diversity as well. So these were the types of pollination. Cool? I hope you're understanding. If you're understanding, do let me know. And also please drink enough amount of water. I'll also just take a break and drink a little amount of water. Too much of talking, right? At least this much. Okay. So now if we talk about agents of pollination. Now pollination, obviously you need pollinators unless and until it is closed flowers, as you saw over here in the case of clistogamous flowers, that is oxalis, viola and commelina, they have it. Okay. Now going ahead, Wind pollination. There are different agents of pollination that we need. First one is wind pollination. Before that, if you categorize agents of pollination, you have biotic and abiotic. Biotic as in the living ones. For example, insects, animals and things like that. Abiotic as in the non-living ones such as wind, water and things. What is more common? Biotic ones are more common because you cannot basically rely on abiotic things, but still abiotic things generally happens as well. So let's see. So in all of them, let's see what is the most common thing. So in the case of abiotic, wind pollination is the most common thing, okay, and where do you find it? So it is common in grasses, my dear children. Here, the scarcest of pollen grains you should be remembering. So pollen grains, they are light and non-sticky for easy wind transport. Obviously, it has to get transported. So if it is very big and things like that, can it be transported? No. Talking about the stamens which are there, okay, the stamens which are there that carries the pollen grains and everything, it has to be well exposed so that whenever wind comes, it can carry along all the pollen grains. Talking about the stigma, it should be what? Large and feathery. It should be large and feathery so that it can trap or any pollen grain that is coming along the way. Flowers, if we talk about that, many are there but with single ovule per ovary. Okay, because wind pollination is not assured that pakka it will happen. So if you make multiple ovaries, it will be a loss for the plant. So what do? There will be so many flowers, but each of the flowers will be having single ovule only. So you should be knowing about all this thing. And the example is corn-cob tassels, my dear children. So over here you see this is an example of wind pollination. Again, pollen grains are light and non-sticky. It should not stick. If it sticks, it cannot move around. Stamen, my dear children, they are very well exposed. Talking about the stigma, they are feathery so that they can trap things. An example, as I said, is corn-cob tassels and also grasses. So if we are asked what is the most common type of pollination in the case of grasses, it is wind pollination. The question has come. Now, agents of pollination, that is water. Now, hydrophyly you call it. Now, rare in flowering plants, Certigenera, mostly monocots are the ones that show this. Common in algae, bryophytes and pteridophytes, they need water for fertilization. Example is you have Vallisneria and Hydrilla and Zoostera as well. Vallisneria, Hydrilla and Zoostera. Methods, let's see. So, Vallisneria, the female flowers float, the male flowers or pollen drift to them. Okay, they float and they drift on the surface of the water itself in the case of Vallisneria. What about the sea grasses, that is Zoostera? In the case of Zoostera, pollination occurs underwater. The pollen is ribbon-like. The pollen will be inside the water and will be moving. It's not floating, it will be inside. These important details are really good for you to remember. Now, obviously it is in water, it has to be protected. So what do you have? They have mucilaginous covering that prevents what? Wetting. Okay, now there is water lily, lotus and things like that you might have seen, right? Do they show water pollination? No, even though they are in water, they show insect pollination, my dear children. Okay, they have pollinators like bee and everyone that help in transfer. So they show entomophily, that is insect pollination. You do not have to remember the name if you cannot, but just have an idea. Okay, example bees and beetles and everything. So I hope this is done. Vallisneria on the surface, sea grasses within the water, and the pollen grains are ribbon-like. Cool, works. Let's get ahead. Now, agents of pollination, that is animals and insects. So common pollinators are bees, butterflies, flies, beetles, wasp and everyone. And then something that is not common, primates as in lemur, rodents, reptiles as in geckos as well as lizards as well. They also do pollination. Please remember it. Now, adaptations are there. Insect pollinated flowers, they would be large, colorful, fragrant and nectar rich. If there are small flowers, they will come in bunches. They would be clustered in inflorescence, my dear children, for visibility, higher visibility. Fly and the beetle pollinated flowers secrete foul odor because fly and all, they get attracted to foul odor. There will be sticky pollen ensuring attachment to animal body. So always use logic. Animal is coming over here and sitting on the plant for having nectar. If the pollen grains are not sticky, it won't be sticking. What is the purpose? Basically, this particular guy who is coming for the nectar, we should be going to another flower, and when it goes to have the nectar there, it should be transferring the pollen grains. So the pollen grain which are there, it should be sticky, my dear children. Okay, talking about the flowers, if they are small, they should be in inflorescence, very important feature. They should be large, colorful and should be fragrant. And if it is done by fly or beetle, my dear children, it should secrete foul odor. Okay, now there are certain rewards also that the plant gives, that is nectar and pollen attract. So nectar and pollens which are there it is a reward. Apart from that, there is also one more reward, that is to provide safe space for egg laying. You know, the big flower that is Amorphophallus, it provides a safe space for the egg laying of all the pollinators which are there. Now, this is specialized relation which is being mentioned in your book, that is about yucca and the moth. Yucca and the moth. So, yucca is a plant and moth is obviously an insect. When the insect come for for nectar and things like that, what happens is that yucca will be transferring its pollen grains. But it's not just that. Yucca will be providing a space so that the moth can come lay its egg and basically help provide a good atmosphere over here. Okay, and obviously with the help of this, yucca is able to do the pollination as well. Not the yucca is able to do the pollination, but it is able to transfer its pollen grains to other plants for reproduction to happen. So, yucca is a plant and moth is an animal. Animal as an insect. Please remember that. Okay, cool. Done. My energy is going low. Okay, now we have certain outbreeding devices. What do you mean by outbreeding devices, my dear children? Such that you saw in autogamy always there is self-pollination happening. But what is the problem of this? Whenever self-pollination happens for a longer time, there will be inbreeding depression, my dear children, because of continuous self-pollination which is happening. And to prevent that, the plant itself has come up with certain techniques such as the time difference. So, pollen release and stigma receptivity would not be synchronized. So, if it's not synchronized, obviously the pollen grain comes and lands, still it won't be able to germinate. Now, there is spatial separation. Anther and the stigma would be positioned differently. Okay, they are positioned differently. For example, anther might be here, stigma might be at top. Obviously, this cannot go fly up, right? So, spatial separation is there. Now, something that plants generally have self-adapted is self-incompatibility. That is genetic mechanism. This is the only genetic mechanism that prevents self-pollination from fertilizing ovules, my dear children. So, genetic mechanism as in, even the pollen grain comes and land, it won't be able to do the proper germination over there because it is incompatible. Lastly, you have unisexual flowers, as in monoecious flowers such as castor and maize. Male and the female flowers on the same plant. Okay, so over here what happens is that, suppose these have the female part, another flower will be there, it will be having only the male part. So obviously you are reducing self-pollination to an extent because you do not have the male part over here. But okay, so I will say that autogamy is prevented. Autogamy is prevented for sure. But what about geitonogamy? From here to here, still it can come. So, geitonogamy is not prevented, even in monoecious flowers. So, castor and maize is an example of the CM, Chief Minister. Chief Minister and the bodyguards, whatever you do, they will be there still there. You can remember like that, or however you want to remember, you remember. Now, dioecious as in papaya, where there are two plants. In one plant there will be only female flowers, in another plant there will be only male flower. Obviously in this case geitonogamy is prevented, autogamy is prevented, xenogamy can take place, and that's totally fine because you are talking about two different plants. So dioecious as in papaya, male and the female flowers on different plants prevents both autogamy and geitonogamy, and this is perfect. Okay, so please remember the name castor, maize and papaya, very important for your KCET. Going ahead. Now, there is something that is known as artificial hybridization. So, I generally cannot depend on plant for all this thing. Again, self-pollination can happen, may not happen. So, what do? Basically, we have artificial hybridization where I know that this is a plant I want, this is a plant I want. Basically, I want to create a hybrid of it. So, what do? I am going to cross different species genera for superior commercial varieties. First thing that I do is emasculation. What do you mean by emasculation? That is, suppose this is a bisexual flower. Okay, this is a bisexual flower, as in it has both the sexes together in one particular flower itself. So, over here you saw this is unisexual flowers. This plant is monoecious because it has both the sexes together, but over here this is unisexual flower. There can be another case where both the flowers has both the thing. Still it will be monoecious because both the sexes are on the same plant. Okay, and the flowers are bisexual. So, in monoecious you have bisexual flowers or unisexual flowers. Cool. Now, over here, suppose it's bisexual, my dear children, what do? You remove this anther so that this anther does not produce pollen grains over here. This is known as emasculation. Removal of anthers from bisexual flowers before dehiscence happens using the forceps that is there. Now, obviously, I have to make sure my stigma is still being really safe, or my female reproductive part is still being safe. So, what do? I cover it with something, and this process is known as bagging. I will wait for the stigma to be receptive, and then what will I do? I'll remove the bag and slowly put my pollen grains which I am looking for. There's no plant that I want. You get it? So bagging is done, that is covering the flowers with butter paper to prevent unwanted pollen contamination. Then pollination process, once the stigma is receptive, desired pollen is dusted as I said, and the flowers are rebagged so that the fruit development take place. Now for unisexual flowers, no emasculation is needed. So this was in the case of bisexual flowers when you had both the sexes together. If it is a unisexual flower, do you need emasculation, my dear children? No, you just need bagging. You just need bagging, no need of emasculation. Now this process is known as hybridization, artificial hybridization. Cool? Works? Wait. I did something. Okay. There's a point, it has been missed. Chalo. Now, basically, we are going to talk about, okay, this slide you can take a screenshot or basically in the PDF anyway you'll get. That is something that is known as double fertilization. What do you mean by that? Again, so you have the stigma, the style and the ovary. Inside the ovary, ovule is there. Inside the ovule, what do you have? You have the female gametophyte. That is the embryo sac. Okay. In the embryo sac, you have the egg cell. This is the egg. And there are two synergids over here. And there are three antipodal cells over here. And there is what? Two polar nuclei over here. The male gamete will be coming and landing over here. Only if this chemical composition everything is okay, that's when it will be forming a pollen tube. It will be germinating and forming a pollen tube. Now this pollen tube, my dear children, as I told, it will always consist of two male gametes. How many? Two male gametes. Listen to me carefully. Okay. All the cells in the female gametophyte are haploid. If you remember, all of them, 1, 2, 3, 4, 5, 6, all of them are haploid. This is haploid nucleus, this is haploid nucleus. Cool, because you got it from the, you got it from the megaspore, right? From the megaspore, the functional megaspore is the one that forms the female gametophyte. So functional one was haploid. So all of the nucleus, it has haploid content. The male gamete, one of it, the first male gamete, will be coming and fusing with the female gamete over here, but it's not direct. The filiform apparatus of the synergids would help in the direction and would help it come and attach. So basically I will say it comes and fuses with the egg, and this is known as syngamy. We call syngamy, but what is more important is that this produces zygote. This produces what? Zygote. Now you have the second female gamete. Second male gamete. Second male gamete and fuses with the two polar nuclei right over here. Yes, you heard me correctly. So it is two polar nuclei that it binds with. And this is known as triple fusion. Why do I call it triple fusion? One male gamete plus two polar nuclei, three nucleus are being fusing over here, which is why I'll call it triple fusion. And what do I get at the end of it? What do I get at the end of it? I get primary endosperm nucleus. I get primary endosperm nucleus, or PEN. If I talk about the cell that is central cell, then I can call it now primary endosperm cell as well. Cool. Over here, how many times is the male gamete fertilizing or fusing? This is the first one and this is the second time. So I'll call this as double fertilization which is happening. Now always remember this primary endosperm nucleus, wait, before that. The male gamete was having haploid content of DNA, right? Okay, it was coming from pollen grains, right? And egg also has haploid. What about zygote? N and N comes together, it becomes diploid. It becomes diploid, that is 2N amount of chromosome is there. Here, male gamete has N, the two polar nuclei, each of them has N. So 1, 2, 3. This will be triploid endosperm, my dear children. Please remember, it will be triploid endosperm. Now I'm going to tell you one more very important thing. The endosperm which is there, it will be, forming before the zygote. Okay, it will be dividing itself and forming before the zygote. Why? Because the endosperm is the one that will be providing nourishment to the zygote to form into an embryo. This zygote is single celled, it has to divide into an embryo, right? And for that you need to provide nourishment. Who provides it? The primary endosperm nucleus. Got it? Or basically the cell. So this is about double fertilization. Going ahead, endosperm. Talking about that. So why does endosperm develop first? I told you it provides nutrition to the developing embryo. Now there is primary endosperm nucleus which divides repeatedly, the nucleus which is there, it will divide repeatedly to form the triploid endosperm tissue. So it will be forming triploid endosperm tissue, as in there will be so many cells, all of them having 3n amount of chromosome. Now, free nuclear endosperm is there. PEN undergoes nuclear division without cell wall formation. So sometimes what will happen, the nucleus is there, it is not going to form cell wall. So some of it will be in free condition like this. Okay, this is known as free nuclear endosperm. Example, the coconut water you drink, it's nothing but the free nuclear endosperm which is there. Now later, slowly, it will be cellularized, and when it becomes cell, my dear children, it will form the white kernel that you eat. Okay. Now, the fate of the endosperm. What is the fate of the endosperm? How is it going to be utilized? What is it going to be in future? So, you know it is there for providing nourishment to the zygote, so that the zygote can be transformed into an embryo. So, there are certain plants which will use the endosperm completely. They will consume it before the seed maturation happens, and the plants are pea, groundnut and beans. I'll tell you one important thing here itself. These plants are known as dicotyledons. What do you mean by dicotyledons? So, zygote to embryo formation, when it happens, what does the embryo do is that it will utilize all the endosperm and convert into another source of nutrient that is known as cotyledon. So it says I don't want the endosperm, I'll make my own nutrition thing, that is cotyledons. So basically it utilizes all the endosperm to form two cotyledons, which is why it is known as dicots. So when you break off a pea, you can break into two, groundnut also two, beans also two, because all of them they are dicots. So that means whenever you have dicots, they have utilized all the endosperm over there, my dear children. Okay, now these plants are also known as, or these seeds are also known as non-albuminous seeds. These seeds are known as non-albuminous seeds, where it is utilizing everything. Okay, now persistent mature embryos, in somewhere the endosperm is still persisting. For example, castor, coconut, etc. Castor is an example of monoecious plant as well, remember that. Now over here castor and coconut is in there, the endosperm still persists. Okay, and what do I call them? I call them monocots, my dear children. In them, they do not make two cotyledons, they just make one cotyledon, and they will be having some amount of endosperm still left. So I cannot break them into two, they just have one cotyledon, that is castor, coconut and everyone. I want you to remember in this way because you might be able to relate it and learn better. Okay, and these plants are known, these seeds are known as albuminous seeds. So whenever the ovule, my dear children, I said all those things, fertilization, everything was happening. At this point of time, the ovule which was undergoing fertilization, that is the male and the female getting, what happens to the ovule? I will call this as a seed now, because inside the seed I have the zygote which will be forming the embryo. What about the fruit? Basically the ovary which is there, it will turn into a fruit. It will be turning into a fruit. And now we are going to talk about, we spoke about endosperm, now we are going to talk about the embryo which is there. So location develops at the micropylar end because the egg was there. Okay, adaptation is that zygote divides after endosperm formation to ensure nutrition. Okay, in the zygote stage, the embryogeny stage, the zygote, it will transform into pro-embryo, then to globular stage, then to heart-shaped stage, then to mature embryo. These steps you have to remember: zygote to pro-embryo, then globular stage, then heart-shaped stage, then mature stage. Okay, over here, zygote, this is pro-embryo, this is globular embryo, this is heart-shaped, and this is mature stage that you get of the embryo. Now, you have two type of seeds, I told you, dicots and monocots. So, you have two type of embryo as well. Okay, in the case of dicot, I'll tell you how it works, and in the case of monocot, also I'll tell you how it works. For that, I have to make a diagram. Okay, see, so there's an embryonal axis. Okay, now, this thing which is there, my dear children, this is the embryonal axis. At the end over here, you have the things that will be forming the roots. Okay, so I'll call this region as radicle. And this region is going to form the shoot, which is why I'll call it as a plumule. Okay, here to cover the root, to keep it safe, you have root cap. I am talking about a dicot embryo. Okay, what happens in a dicot seed? Basically, now I told you the endosperm makes the cotyledons. Endosperm is being utilized to make the cotyledons. So they do have two cotyledons which will be like this. So they do have two cotyledons, something like this. In your NCERT, it's been mentioned like this. And here the plumule is mentioned. Just a second. Okay, fine, it's okay. Here, this part, the extra part which is there, I have made it an upper side. Okay, so this is a plumule which will be forming the shoot, but I have made it down this side. And these are the two cotyledons which are there. Cool. Now we'll read it. So embryonal axis with two cotyledons, epicotyl is there, that is above the cotyledons, my dear children. I told you the above the cotyledons, what do you have? Epicotyl. It will be ending in plumule, that is the stem tip. There is hypocotyl region, that is below the cotyledons. Okay, so for example, this region which is there above the cotyledons, okay, this is suppose a cotyledon. Above the cotyledon, if it's there, this is known as epicotyl. Below the cotyledon is known as hypocotyl. Hypocotyl basically has the radicle or root tip at the end, and the root tip is covered by the root cap. Talking about the monocot embryo, things are quite different there. They just have one cotyledon, and they also need to have what? They also need to have this thing. What do you call it? Endosperm. Yes. Oh my god. So I kind of zoned out while teaching. Oh my god, will that happen? Can that happen? So just listen to me. So again, you have an embryonal axis over here. Okay, my dear children. Now, you have one cotyledon over here which you call as scutellum. Okay, what do you call it as? Scutellum. It has a specific name. And it also has some amount of endosperm still persisting. And this particular region, as I said again, this will be plumule. This is known as radicle. Okay. Now, there is one very important thing, that is the plumule is covered by certain things which is known as coleoptile. And this is going to be forming the leaves and everything out there. Okay, it's a leaf primordia. Over here, the radicle is covered by root cap for sure. But beyond it, it also has something else which is known as coleorhiza. So this is how the structure of monocot and dicot embryo will be differing. So scutellum and little amount of endosperm still persisting. So one cotyledon, that is scutellum, lateral to the embryonal axis, one side of the embryonal axis. Radicle and root cap enclosed in coleorhiza, and epicotyl with shoot apex and leaf primordia inside the coleoptile which is there. Cool. And this is all the structure of the embryo which is there. Talking about the seed in general, we spoke about the embryonal axis and everything. Now in the seed you have the embryo, right? In the seed you have the embryo. So let's see, the final product of sexual reproduction is what? The fertilized ovule inside the fruit. The seed structure, it will be having a seed coat. Okay, it will be having a seed coat. So basically the ovule integuments which were, the ovule is now becoming a seed. So the integuments which are there, it will be acting as a seed coat. What is it? It is testa and tegmen. It will be now known as testa and tegmen, my dear children. So it will be there will be seed coat. Apart from that, cotyledons will be there, either one or two. Now embryonal axis will be there, that is basically talking about the embryo. Inside the seed coat is there, cotyledon is there which will provide nourishment to the embryo, and the embryo will obviously be there. Type of the seeds, there are non-albuminous and there is albuminous. I told you, whenever the endosperm is completely used, it means that is non-albuminous. For example, pea, groundnut, all the dicots come under this. Albuminous as in when the endosperm is partially remaining over there, such as wheat, maize, barley and castor. Okay, same thing, monocots. Now there is something that is known as perisperm. What do you mean by that? So remember, when we were talking about the nucellus and everything, okay, we were talking about the ovary. Inside the ovary there was ovule, inside the ovule there was megasporangium, inside the megasporangium you had nucellus. One cell of the nucellus was forming in the embryo sac. What about the other cells? Other cells usually they get lost, they get degenerated, they get used up. But in some they persist. In some they persist and they provide nourishment. You call this as perisperm. Do not get confused with pericarp. Pericarp is the covering of the, what do you call, the fruit which is there. Pericarp is different. You're talking about perisperm. Perisperm is what? When the persistent nucleus which is there, it is still persisting, you will call this as perisperm. So example is black pepper and beet. Black pepper and beet is an example of this. Now micropyle which is there, small pore. Okay, so in this ovule also, that is a seed, my dear children. In the seed, you will be finding a region which has a small pore, that is micropyle, and the small pore is for oxygen and water entry during the germination of the seed. The seed will germinate, and whatever things are there, it will get into a seedling and things like that. Okay, seed maturity. So water loss 10 to 15% happens, it becomes dry. Dormancy or germination with moisture, oxygen, temperature, slowly it will become germinate, or if at all it's not there, it will be in the dormant state. This is all about the seed. Okay, please do not forget the perisperm and also the example, that is beet and black pepper. Both of them, they start with B and B. Cool. Going ahead, advantages of seed. Okay, and the seed longevity. So advantage of the seed is what? Independence from water. Pollination and fertilization do not rely on water, my dear children. Okay, and that's not an advantage of the seed. Okay, I'll remove that point. Okay, dispersal. Seeds help plant spread to new habitats. That is obviously a very important thing. Obviously, sometimes we cannot take the plant as a whole, but the seed, when it disperses here and there, okay, it will obviously help in the dispersion of the plants which are there. Now, food reserve nourishes the seedling until they start photosynthesis, protection also, and provides genetic variation as well. Now, seed longevity. Some seeds lose viability in months and some will be lasting for years. So, oldest viable seeds, my dear children, very important question. Lupinus articus, okay, 10,000 years old. It was from Arctic tundra. 10,000 years old seed, my dear children, from Lupinus. The plant's name is Lupinus articus. Then you have Phoenix dactylifera, date palm, okay, that remained viable for 2,000 years and it was excavated from Dead Sea. Okay, when King Herod's time was there at that point of time. So Lupinus articus 10,000 years and Phoenix dactylifera 2,000 years. Cool. Will you remember this? Yes. Now, fruit formation. Let's see about fruit formation. So we spoke about the ovule and the seed and everything up there. I told you the ovary which is there, it is going to form the fruit. So ovary, fruit also will be having certain covering and things like that. Let's see. So ovary is the fruit and pericarp forms the fruit wall. So in the ovary, my dear children, this outermost thing, suppose this is, let's suppose this is mango. So over here, inside the mango, you know there is seed. Seed will be having seed coat and everything. If you talk about mango, this is an ovary. Ovaries structure, the wall is what? It is known as pericarp. Pericarp basically is divided into different things. Outermost layer is epicarp. Then you have middle layer, the fleshy thing that we eat is mesocarp. And then, you know, gutli, or basically the innermost hard structure which is there, inside which the seed is there, my dear children, you call this as endocarp. You call this as endocarp. Cool. All of them, they form the pericarp. Now, there are some true fruits in where the ovary is the one that will be forming structure like this, that is, it will be giving rise to fruits. But there are false fruit also, where it's not the ovary. Apart from ovary, if anything else gives rise to fruit, it is known as false fruits. For example, you know, flowers, how do they look? So flower petals are there, arranged on thalamus regions, my dear children. Suppose this thalamus is also helping in the formation of fruit, I will call this as a false fruit. And these things happen in apple, strawberry and cashew. Okay, I'm not saying this is a thalamus that will be growing into fruit, but in them, things apart from ovary are contributing to the formation of fruit, which is why apple, strawberry and cashew, they are known as false fruits. So over here, if we see the structure of apple, okay, there was a recent question how to label it. So this is the seed that is there inside, okay, and then you have multiple layers. So seed, and then you have the endocarp, then you have the mesocarp, and then you have the epicarp here inside the seed only, inside the fruit only. Now this layer that is there, it might be the thalamus from which it has been formed. This is the thalamus from which it had been formed. Every other part of the ovary is here and the ovule will be over here. Got it? So thalamus is also contributing over here. Now there is parthenocarpic fruits. What do you mean by that? That is a totally separate thing. Okay, what do you mean by parthenocarpic fruit? So develop without fertilization. So over here usually we said that ovary, basically when the fertilization happens, the ovule becomes seeds and the ovary becomes fruit. But over here in the case of parthenocarpic fruits, my dear children, without fertilization, the ovary is still becoming a fruit. And why does this happen? Probably because of the hormones which are there. Some plants, some hormones are being released, which is why it will slowly transform into a fruit. Okay, without fertilization. Now this can be natural or this can be induced. Banana is an example of this, where it happens naturally. It can be induced as well. If I put growth hormones into the ovary, it won't need fertilization for the fruit formation to happen. Without fertilization, when the fruit formation happens, you call it parthenocarpic fruit. So seedless variety of fruits can be obtained from this. Always remember, parthenocarpy is different from what you are going to learn now, that is apomixis. In the case of parthenocarpy, you are making a fruit without fertilization. In the case of apomixis that you are going to study now, you are going to make seed without fertilization. That is kind of very, very tricky, and this is the last part of this particular chapter. I hope I have explained each and everything to you in depth, in detail, in a very short format. So do let me know if it helps in the comment section, and do let me know what all doubts have come to your mind in the comment section. Again, I will be solving each of your doubts. Cool? Now over here in the case of apomixis, my dear children, the seed formation occurs without fertilization. This is very common in Asteraceae and grasses. Okay, so what will happen? I'm telling you. This is the embryo sac, the ovule, right? There is the egg cell and the synergids and the antipodals and the polar nuclei. Here the egg cell somehow it is going to form a zygote. It is going to form a zygote without the need of a male gamete. And when it forms a zygote, now I'll be calling this as a seed. So it basically mimics sexual reproduction, but it is actually a form of asexual reproduction. Why? Because only the female gamete is being taken. That means the female plant characteristics is being seen in the progeny which will be produced in the seed. There is no other parent, there is no genetic variation. So it is asexual reproduction, there is no fertilization of gametes. Now types: diploid egg cell forms without reduction division, develops into an embryo without fertilization. Okay, so there are two reasons how it can happen. Okay, the egg cell which is there, my dear children, it might have formed. Whenever it was formed in diploid state only. It didn't undergo all the divisions to form haploid. It was diploid, and this diploid egg cell which is there, it is going to give rise to zygote, because you know zygote is also diploid. This is a process that happens. Okay. Now in other cases what happens, the nucellar cells which are left behind, my dear children, a lot of it will come inside. Will be coming inside. And nuclear cells, my dear children, again they were normal cells. They didn't undergo meiosis and all. So they were again 2n. So they come inside, and that's when another condition arises, that is you get multiple embryos. You call this as polyembryony. First thing, okay, listen, whenever a seed is being formed without fertilization, you will call this apomixis. Now this can happen in two ways. One way is that the egg cell which was there, it was already diploid, it was not haploid. So it does not need another male gamete to make a diploid zygote. It itself is diploid, so it can just make a zygote. Okay, that is one case. Second thing, the nuclear cells which are there, it can get inside, and when these cells get inside, they are going to form multiple embryos. Okay, they are going to form multiple embryos. So I'll call this condition as polyembryony. Both these conditions can happen. So about the nuclear cell that I was talking about, it happens in the citrus and the mango plants. All the citric varieties of plants, on the mango plants, this particular thing happens. Now polyembryony is when more than one embryo per seed occurs. That is example in the case of orange. You will be seeing multiple seeds. You will be seeing multiple embryo per seed which is there. Okay, you won't be seeing multiple seeds. Okay, multiple embryo per seed which is there. Now Genetic nature. Apomictic embryos are clones. Why do I call it clones? Because from the female parent, there is no male parent, so obviously same like the female one. Now, importance in hybrid crops is that hybrid loses traits if grown from normal seeds. For example, I have a hybrid plant. Okay, and when it will fertilize with another plant, probably it will be losing the character that it was having, because there is new genetic varieties that is happening over here. But if I have an apomictic seed, my dear children, the hybrid varieties are being maintained over here. This is one particular parent plant. Do you need another parent? No. So that is one thing. Now, apomixis prevents segregation, allowing farmers to reuse hybrid seeds yearly. So there is no point of segregation. Nothing happens. What characters was there initially will be there until the end. It will keep on dividing like that. Now, ongoing research to transfer apomixis trait to hybrids is also been seen over here, but these are the importance that you see. So, in the case of hybrid cross, it becomes, for buying hybrid cross, it is very costly for the farmers. So, if they rely on apomixis, it becomes easy for them. Okay, so if I have a plant, I have all the good characters in that particular plant, I can basically make the plant into apomictic plant with the help of hormones and stuff like that. Okay, and it is generally naturally happening. Okay, it is generally naturally happening, but people are working on it such that the normal plants which are there can be also turned into apomictic, such that whatever important characters are there can be, seen in every single generation. So that is about apomixis. Always remember, apomixis is seed formation without fertilization, and parthenocarpy is basically fruit formation without fertilization. Do not get confused. And with this, we are completing our chapter, that is sexual reproduction in flowering plants, my dear little children. So do let me know if it was helpful for you, and do not forget to like, share and subscribe to the little channel, and please don't worry, everything is going to be really great. You are going to do really good in your exams. Bye bye. Take care.